Students, title your notes allele frequencies and Hardy-Weinberg equilibrium. Hardy-Weinberg is capitalized because it's a proper noun. These are the names of the scientists that kind of quantified this stuff. But you're going to see that this equation that you get, this Hardy-Weinberg equation, is nothing but probabilities. So that's why we have this bead analogy, because if you're drawing a bead randomly from the cup, there's a 50-50 chance that you're going to draw green instead of pink. All right, but let's define our terms, because I'm going to start using the word population. And I want you to know, because there's something specific that I mean when I say that word. And what I'm referring to is a group of individuals of the same species in the same area and are interbreeding. Okay, so those three things would be necessary prerequisites in order to be considered a population. And then an allele is this is actually kind of a refresher of Unit 5, but an allele is just a version of a gene, right? And then so then if you kind of put the two terms together, the gene pool would just be referring to the alleles in the population. Okay, so now we know what we're talking about when I say gene pool. If I refer to the population, you'll know what I mean. So the snails, talking about the guppy population over there, the snails, yes, the snails are in there with the guppies, but they're not part of the gene pool. The gene pool is just the guppies that are breeding with each other. And if I had a molly in there or a goldfish, even though it's a fish, it wouldn't count as part of the population. It's a different species. So again, same species, same area, and are interbreeding. Okay, so now we want to make sure that we make this clear. For evolution to occur, we have to meet some specific criteria. So for evolution to occur, there must be a change in the allele frequencies. If evolution is not occurring, and I'm going to underline this sentence because this is the most important sentence. If evolution is not occurring, allele frequencies will come to Hardy-Weinberg equilibrium. Okay, so underline this. And we can say over time if we want, just to, because I want you to know that it does take time for things to reach equilibrium. But if enough time passes and conditions are right, these allele frequencies will kind of, they will find this equilibrium at which they kind of stabilize the numbers, you could say. So this is all very abstract right now, I know, but I'm going to make it more concrete for you. So there are some conditions, however. So for Hardy-Weinberg equilibrium to be reached, 5 conditions must be met. Now, here's what's kind of interesting. Very rarely, in fact, maybe never, maybe even never, are these 5 conditions perfectly met in nature. So this we're going to say that this represents an ideal, I guess you could say, an ideal that is rarely met. It's more hypothetical, in other words, than it is real. But it is certainly true that if these 5 conditions are present, then the Hardy-Weinberg equilibrium is mathematically true. Okay, you're all so confused right now. It's okay. It's gonna become clear. So let's talk about those 5 conditions, though. Okay. Here's what we mean, right? If you're thinking of the population as a gene pool, and that gene pool consists of alleles, we can't have, like, if we're going to represent this mathematically, what is the probability that when I draw these and I make offspring, what is the probability that I'm going to have, say, homozygous dominant? This is actually heterozygous, right? Now, what are the chances that I draw, okay, another heterozygous? Oh, okay, so now we got homozygous recessive. Line again, because each of the pink is capital P, green is lowercase p, whatever. You can set it however you want. Another heterozygous. But then I could draw randomly double green, homozygous dominant. Right? What are the mathematical chances of this, and how will what proportions would we draw if we did this a million times, right? There's a way to calculate it. It's just probability. And that's what we're about to show you. But what we have to say is that in order for these mathematical principles to apply, it's a very idyllic conditions, right? And usually in nature, there's stuff messing this up. Like in nature, maybe a predator comes in, wipes out half of the population randomly, and then we've skewed the numbers. So in nature, there's things that usually mess it up. But in an ideal situation, if everything is untouched, it will obey the Hardy-Weinberg ratios. So what are those idyllic conditions? In those idyllic conditions, there would be no mutation, which is pretty much never the case. There would be completely random mating. Usually mates are selective, so it's not usually random, right? And usually there is mutation. So I'm showing you that these are very idyllic conditions. Number 3, there's no natural selection. Well, normally there is natural selection. Normally there's a struggle to survive. Right? So, man, how do we ever meet these conditions? 4, you also have to have a very large sample size. And this is to eliminate genetic drift. We'll talk about genetic drift in a little bit later. And then the last condition is that we need to have no migration. So you can't have any like other individuals moving into the gene pool, contributing foreign genes into this gene pool. Because if you did that, well, then you would have gene flow, and you'd have like new alleles brought in that skews that skew the ratios, right? So obviously we have to, that would be like when you're doing your experiment today with these beads, that would be like if somebody just like came over here and just was like, oh, and poured a bunch of alleles into your cup. You're like, hey, you messed up my numbers. I was doing Hardy-Weinberg and now I can't. I messed it up. That's what that would be like. Gene flow, or gene flow from migration. Other populations coming in, bringing in new alleles, and therefore changing the ratios of those alleles that are present. Okay, so if these 5 conditions are met, and we have control over these 5 conditions today, because we have a cup and we have beads, and we can totally mimic these 5 conditions. We're not going to be adding anything in there. We're not going to be mutating these alleles. They're going to stay the same color that they are. We're not, and so we can very easily meet these conditions and simulate Hardy-Weinberg. Okay. All right, so let's okay. So why then would we use Hardy-Weinberg if those conditions are never met? Right? I mean, you guys are wondering that, I'm sure. Why then would we use Hardy-Weinberg if those conditions are never met? And the reason is it gives us a perfect null hypothesis. And I'll tell you why. It gives us a great null hypothesis. So we can detect if evolution is occurring. Because you remember that the null hypothesis, what a null hypothesis is it basically states that nothing is happening. There's no difference between the groups. It gives us a great null hypothesis so we can detect if evolution is occurring in a population. So let's write down our null hypothesis, and we'll also write down the alternative hypothesis. So null just means no difference. There's nothing occurring. So the null hypothesis would say this. You would say the population's allele frequencies are at Hardy-Weinberg ratios, I guess you could say. So therefore, no evolution is occurring. That's the null hypothesis. So the alternative would be that evolution is occurring. By the way, students, this is always your null hypothesis. So any question you get on the test or during the lab, and you're like, shoot, what should I put for the null hypothesis? It's always this. It's always Hardy-Weinberg. Null hypothesis is Hardy-Weinberg ratios, right? And what you're going to see is whether or not you can reject that null hypothesis. And because if you reject it, you are proving that evolution is occurring, because we're finding ratios that are not what we would normally expect. Therefore, evolution would be occurring. So alternative is the population's allele frequencies are not at Hardy-Weinberg ratios. Therefore, evolution is occurring. Or you could say evolution must be occurring. So this is kind of like the chi-squared. You guys remember? So if you thought you were done with the chi-squared, you think again. It's coming back to haunt you again, chi-squared. But maybe you were good at chi-squared, and so you're ready to apply it to a new situation. This situation, question is, what you're gonna see, right? You're gonna have these idyllic Hardy-Weinberg ratios, and then you're gonna compare your actual ratios, and if they match up, do they match up enough in order for us to say that evolution is not occurring? We see some differences in the ratios. Are the differences significant enough to say, yes, evolution must be occurring? How do we know? Chi-squared. Chi-squared is the only way to really detect. So here we go. Okay, this is the analogy. So we have a cup or some kind of container filled with black and white beads. We have 10 black beads. 1, 2. You guys will have actual colors, but my pen is black and white, so we're just using those colors. So inside this container are 10 black beads. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then I have 10 white beads. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right. So, and they're all mixed in there randomly. By the way, you guys are quite literally going to be doing this in just a second with the cups. But the question that I have is, what are the odds that you draw a black bead when you reach in? You're going to draw one bead. Anybody know? So if you got 10 black alleles, by the way, the black allele is in the Hardy-Weinberg equation, we denote it as P. So we're going to be using P and Q. So the black allele is going to represent P. And inside the container, we have a ratio of how many total beads do we have? 20, right? 20 total. And how many of them are the black allele? 10. 10 out of 20 is the same as a 0.5 frequency. And so so that equals P, okay? So P equals 0.5. Actually, let's write it maybe. Let's just write it like this. P equals 0.5. All right, so now for the other allele, which is the white allele, which is represented by Q. And this might already be confusing you because you're like, wait, I thought it would be capital P or lowercase p. The Hardy-Weinberg equation just uses P and Q. That's the format that they're going to use. Okay, so just try to get used to that. But let's say that we have 10 out of 20. Again, the other 10 are white alleles. Then that means that the frequency of Q is also 0.5. All right, so students, this little container here represents the gene pool, but it also kind of represents what we're going to call a mating chamber. And so by randomly draw 2 beads to determine the phenotype, or the genotype, I should say, of the offspring. So if we're going to make an offspring, we're going to have to randomly draw alleles from the container to make those offspring. Okay, so here's what I want to ask you guys. What are all the possible genotypes that we could have? We have P and Q. We know that an individual has to have two alleles, right? So what are the possibilities? Somebody, this is not rhetorical. I'm hoping you can help me out here. Yeah.
Like all of them?
Yeah, there's only three possibilities. Because each individual has to have two alleles. Anya? Double P, PQ, and QQ. Those are the only possibilities, right? Everybody good with that? Somebody might be like, wait, QP. That's the same. QP and PQ are the same. Okay, so there's only three possibilities. You guys saw that. I drew out the beads. We got double purple, double pink, and then pink and purple. There's three possibilities. If we were to calculate the odds of double P, how would you calculate those odds? Okay, so here's how you think about it, right? I've got 10 pink beads and 10 green beads. What are the chances that the first allele is going to be green? 0.5. What are the chances that the next allele is going to be green? Okay, so the law of probability says that you have to multiply the 1/2 chance times the 1/2 chance. And if you do that, what you get? 1/2 times 1/2 is 1/4. So 0.25% chance. And look, we did it. 1 out of 4 chance. So let's show that real quick. Remember that it's 1/2 chance for the first P times 1/2 chance for the second P allele equals 1/4, which means that P squared equals 0.25. Why P squared? Because it's the probability of P times itself. P squared. Okay? Everybody okay with that? Logically, mathematically, we all agree. All right, what would be,
So I had a question about the second half. Are we counting that without substitution? Because wouldn't that be 9 on 20 then? Because once you've taken a green out of the pool, or do we just count it as if there were?
We're pretending there's no substitution.
Oh.
Yeah, that's a good point though. Technically, it wouldn't be. Yeah, I don't know. I'll have to think about that. No one's ever pointed that out. But yeah, you're pretending there's no substitution in this case. This is an ideal situation. PQ. How would we calculate PQ? What are the chances that I draw P? 1/2. What are the chances that I draw a Q right after that? Again, it would be 1/2 times 1/2, right? Here's what's kind of weird about this, though. Does anybody, actually, maybe Anya would be able to tell us. Anya, are we done here? Is the probability of drawing PQ just 1/4? Because what happened here was we drew a P, and then we drew a Q, and that's how we got PQ. Or is there another possibility? Yeah, so the other way it could have happened is we had drawn Q, and then which would be 1/2. And then there's another, but then right after that, you'd have to draw P, which is a 1/2. So this is what we call the law of, we've got the law of multiplication and the law of addition, because there's two possible ways this could happen. So what you have to do is you have to take the sum of these two possibilities. 0.25 plus 0.25 is a 50% chance. So the way that this is denoted in the Hardy-Weinberg equation is 2pq equals 0.5. So why do we have the number 2 in front? The number 2 is just representing that that there's two possible ways to get the heterozygote. Are you okay with that? All right. Now the last one, QQ. Probability of QQ going to be identical to the PP thing. So once again, 1/2 times 1/2 Q squared equals 0.25. All right, so what I want to do now is put it all together. So how could we represent this as one big equation? Does everybody notice what these decimals all add up to? So if you take like whenever you calculate a probability and you say that you exhaust all possibilities, then your probabilities at the end, they should all add up to 100%, right? And if we look at our equation, our percentages, our probabilities do in fact add up to 100%. If they don't, then you did not account for all of the possible outcomes, right? So here's how you represent this as one big equation. You would say p squared plus the 2pq plus the q squared has to equal 1. Does everybody agree with that statement? Mathematically speaking, right? That has to be true. So that is the Hardy-Weinberg equation right there. There is one other mathematical statement we can make, and that is that if you added up all the alleles, so say that you had P plus Q, that would also have to equal 1. Or another way of saying that is that that would have to equal 100% of all the alleles. So these two mathematical statements right here are what you're going to be working with throughout this little simulation that we're about to try. And these are the Hardy-Weinberg equations. And they only apply if the 5 criteria are met, which they will be. Those 5 criteria will be met in our scenario because we've got a controlled environment. We have a cup with beads in it, so there's no way anything can interfere. But in something like an aquarium, well, actually, an aquarium is too controlled environment. But say a pond. Well, a pond, animals can enter into that pond, and you can have gene flow, and that would mess up the ratios of these alleles to where Hardy-Weinberg is not being met. Right? But in a perfectly controlled environment, we've got it dialed in. These probabilities, the law of probability has to, like, our experiment is going to obey those laws of probability. The only way in which it couldn't today, so probably, right, when you guys are working with this today, you're going to produce like, I don't know, I think it's 20 offspring. 20 is kind of a small pool, kind of a small sample size. So remember with the coin when we flipped it like 100 times, and we were like, okay, we should get 50-50, 50 heads, 50 tails. When you're only doing it 100 times, sometimes it can be off by 45, 55, just by chance it can be off. So with a smaller sample size like today, it could get a little bit off of those Hardy-Weinberg ratios, but we know it's going to be close, right? When you're doing the drawing. Any questions about this? About this? Yeah. Okay. Okay. Questions? Yeah. The only other thing I was going to say is, so let's say that when you do the experiment, you find ratios that don't jive with the Hardy-Weinberg numbers. That immediately tells you that something is influencing the result. And so I might ask you, well, what things do you think are influencing the result? And you would go back to this list and you would say, well, maybe there's mutation that's influencing the numbers. Maybe the mating is not random. Like maybe there's some selection going on, mate selection. Or, oh, you know what? Maybe there's natural selection. There might be some there might have been like some real strong environmental challenge that favored one allele over another, right? Or you could say, well, the other thing that could be influencing it is we have a tiny, tiny sample size, and there could be genetic drift going on. Or you'd say, well, maybe there's migration. There could be alleles coming in from another population. So these are all five of these are possible explanations as to why your numbers did not meet the Hardy-Weinberg ideal, if that makes sense. All right, I think you guys are ready to try the simulation.